Titre du mémoire. Diplôme d'université du goût. De la gastronomie et des arts de la table. Dugat. Mémoire de fin d'études. May 2014. Wine tasting and critique. Bias, errors, misconceptions and their influence on the market. José Luis Borges. I. Table of Contents. Abstract. 3. Acknowledgements. 4. Introduction. 1. Anatomy and physiology of the sense organs. 3. 1. Vision. 3. 2. Olfaction. 6. 2.1. Transduction. 8. 2.2. Communication among olfactory sensory cells. 8. 2.3. Integration in the central nervous system. 9. 3. Gustation. 9. 3.1. Transduction. 10. 3.2. Communication among gustatory sensory cells. 12. 3.3. Integration in the central nervous system. 12. 4. Chemosensory trigeminal system. 13. Chemical origin of wine attributes. 14. 1. Color. 14. 2. Aroma. 15. 2.1. Primary aromas. 15. 2.2. Secondary aromas. 18. 2.3. Tertiary aromas. 19. 2.4. Aromatic defects. 20. 2.4.1. Oxidation. 20. 2.4.2. Reduction. 20. 2.4.3. Corky. 21. 2.4.4. Acetic acid and ethyl acetate. 22. I. 2.4.5. Other aromatic defects. 22. 3. Taste and oral sensations. 23. 3.1. Sweetness. 23. 3.2. Acidity. 24. 3.2.1. Acids derived from grapes. 25. 3.2.2. Fermentation-derived acids. 25. 3.2.3. Measurements of acidity. 26. 3.3. Bitterness and astringency. 27. 3.3.1. Non-flavonoids polyphenols. 28. 3.3.2. Flavonoid polyphenols. 29. Wine assessment. 31. 1. Causes of bias and misconceptions in tasting. 32. 1.1 Physiological factors. 32. 1.1.1 Sensory adaptation. 32. 1.1.2 Sensory thresholds and individual variation. 33. 1.1.3 Genetic variation. 34. 1.1.3.1 Vision. 34. 1.1.3.2 Gustation. 34. 1.1.3.3 Olfaction. 35. 1.1.4 Age. 36. 1.1.5 Cross-modal perception. 37. 1.2 Psychological factors. 40. 1.3. Knowledge and training. 41. 1.4. Language. 42. 1.5. Information. 44. Discussion. 46. Conclusions. 54. References. 55. 3. Abstract. Tasting is an important tool for evaluating wines, as it exerts a decision-making role at the various stages of production and distribution chains of consumption. Tasting is also the instrument of wine critics and of wine contest judges. Assessment of specialized critics and the results obtained in wine contests exert great influence over wine market. The outcome of these evaluations, nevertheless, has revealed poor accuracy, precision and reproducibility. Still, the wine market is strongly influenced by such results. This thesis introduces a revision on anatomical and physiological knowledge of the sense organs involved in the sensory evaluation of wines. The chemical basis for the origin of wines' organoleptic characteristics regarding their appearance, aroma and oral sensations is also analyzed. Based on this knowledge, the causes of errors, deviations and inaccuracy in wine tasting are then discussed, as well as how these factors contribute to illusory results at wine contests and to the assessment made by skilled critics, which is often dissociated of consumers' perception. The influence on the market and wine prices of the evaluation performed by specialized critics and awards obtained in contests have been focused in this analysis as well. Some options of wine's assessment are herein proposed, in order to meet the needs of regular consumers. 4. Acknowledgements. I wish to extend my sincere gratitude to the librarians from the Library of the Institute for Education and Research at Hospital Cirio Libans, Adriana Mara Fonseca, and especially to Rita de Casia Ortega Borges for their invaluable collaboration in researching, collecting and organizing the bibliographic references. My special thanks to Noriko La Regina, for composing and formatting the text, as she has been doing in so many other publications in the last 20 years. Last but certainly not least, my gratitude to Carmen Sylvia, my wife, for her unfailing support during these past 35 years, and to whom I dedicate this thesis. 1. Introduction. Sensory analysis is a useful tool in the food and beverage industry for product development, testing quality and establishing marketing strategies. 1, 2, 3. On the other hand, in the wine industry, tasting is the basis of all activities, from the viticultural practice to wine service at table and to its consumption. In the words of Émile Peynaud, tasting is a means of knowledge, and getting to know the wine better improves its production, storage, control and ultimately allows us to better enjoy it. For the world consumption of wine, in both producing and traditional consuming countries, has decreased considerably in the second half of the 20th century. Moreover, new consumer markets, especially Asians, have emerged, importing large quantities of wine. In the last decade, there has been a slight overall increase in consumption. Other recent wine market trends are the relative rise in consumption of quality wines, with a consequent decrease in the consumption of wine in bulk, and the increase in price per unit sold. 5, 6, 7. The expansion of the global market for fine wines has led to a greater accessibility to this product, with the growth in the number of uninformed consumers, or even naive ones. This phenomenon paved the way for the emergence of influential wine critics, whose tasting notes play an important role in the global wine market. 8. In a population of wine buyers, the higher the proportion of naive consumers with respect to 1. Adriano G. Cruz et al. Sensory analysis, relevance for probiotic, and symbiotic product development. Comparev Food SCI Food SAF. 2010, 9, 4358-73. 2. Montserrat Rayuam et al. Sensory Analysis in Quality Control, the gin as an example. Cited 2013 December. Available from http://www.intechopen.com/download/get/type/id/23744. 3. Maria Ionario et al. Sensory Analysis in the Food Industry as a Tool for Marketing Decisions. ADV Data. Anal Classif. 2012, 6, 4303-21. 4. Emile Peynaud and Jacques Bluin. Augusto do Vinho. São Paulo, Martins Fontes, 2010. 5. Glyn Whitware and Jeremy Rothfield. Projecting the World Wine Market from 2003 to 2010. Australas. Agribus Revelation 2005, 13 Paper 21. Cited 2013 December. Available from http://www.agrifood. info/review/2005/Whitware_Rothfield. HTML. 6. International Organization of Vine and Wine. World Vitiviniculture Situation in 2012. In XXXVth World Congress of Vine and Wine Bucharest, 3 June 2013. Cited 2013 December. Available from http://ebookbrowser. net/OIV World Vitiviniculture Situation in 2012 PDF D5026999851. 7. International Organization of Vine and Wine. Statistical Report on World Vitiviniculture 2012. Cited 2013. December. Available from http://www. OIV. int/OIV/info/Enesmeyer OIV Report. 8. Karl Storchman, Alexander Mitterling and Aaron Lee. The Detrimental Effect of Expert Opinion on Price Quality Dispersion Evidence from the Wine Market. AAWE Working Paper. Economics. 2012. 118. Cited 2013 December. Available from http://www. wineeconomics.org/or/WP. Content/uploads/2012/10/AAWE_WP118. PDF. 2. The informed ones, the greater the effect of the scores assigned by critics in pricing. Moreover, a similar effect occurs when a wine receives a second revision grade given by the same critic or another one. In this case, the re-evaluation grade receives a higher weight by consumers, affecting final price. 9. The effect of high grades on the price of the wine seems to be higher than that related to lower grades. 10. However, Inconsistency and poor reproducibility observed in wine competitions. Tasting panels and analysis made by opinion-forming critics lead to questioning the validity of tasting as a method of evaluating wines. 11,12. This thesis has the purpose of reviewing the anatomical and physiological basis of sensory evaluation and the chemical origins of wine organoleptic properties. In addition, the causes of limitations, biases and errors of wine tasting will be discussed as well as their repercussions in the specialized critique of wine and in its market. 9. Michael Gibbs, Michael Tapia and Frederick Warszynski. Globalization, Superstars, and Reputation, Theory and Evidence from the Wine Industry. J. Wine Econom. 2009, 4, 149-64. Available from http://wwineeconomics.org/journal/content/volume 4-2009 no1 10 Hella Haj Ali, Sebastian Lecoq and Michael Visser. The Impact of Gurus, Parker Grades and E.N. Primer Wine Prices. Revised, June 2007, cited 2013 December. Available from http://ams. U Paris 2. fr/doctrave/0718. PDF. 11 David Derbyshire. Wine Tasting, Its Junk Science. The Guardian. The Observer. 2013, the 23rd of June. Cited 2013 December. Available from http://lifeandstyle/20w.com/Jun/23/Wine Tasting Junk Science. Analysis. 12 Richard E. Quant. On Wine Bullshit, Some New Software. J. Wine Econom. 2007 2, 129-35. 3. Anatomy and Physiology of the Sense Organs. The sense organs constitute our interface with the environment. Through them, we perceive the signals of the outside world, which are processed in the upper layers of the brain, thus becoming a cognitive reality. The evolution of such organs has allowed the survival of the human species. Without olfaction, how could one manage to escape in time from a fire in the house or even detect deteriorated food? Vision has allowed us to observe the enemy from afar, and the sense of touch warns us immediately about hot or sharp objects that could cause injuries. The sensorial mechanisms also receive hedonic stimuli. The nerve pathways that carry different sensory stimuli to the cerebral cortex are integrated in an area called the orbitofrontal cortex. This area, in addition to sensory integration, is likely responsible for enhancing affective values, decision-making processes and the mediation of hedonic experience. 13. This integration between vision, olfaction and taste, whose primary biological function is to optimize search for proper foods, can also lead to errors of judgment. Thus, our senses may be misleading, requiring a careful analysis based on experimental knowledge of the information brought by them. The organs of sense, whose anatomy and physiology are described below, are those involved in both hedonic and technical tasting of wines. 1. Vision. The eye is a sophisticated optical instrument developed by natural selection under evolutionary pressures of over millions of years. This sensory organ allows us to capture a range of the light spectrum, the visible radiation, and is one of our important interfaces with the structural organization of the world. The beam of light emitted or reflected by objects goes through the transparent anterior membrane of the eye, the cornea, until it reaches the crystalline lens. This 13 Morton L. Kringel back. The human orbitofrontal cortex, linking reward to hedonic experience. Nat Rev. Neurosci. 2005, 6691-702. 4. Structure resembles a lens and it is deformable by the action of the muscles on which it is fixed. When going through crystalline lens, the light undergoes refraction controlled by the change in the shape of this lens. This process sends the focus of the light beam onto the retina, a nervous tissue located in the posterior part of the eye. 14. The retina is a projection of the central nervous system outside the skull, and it is the only non-invasively observable part of this system. It is a 0.5 mm thick layer, which coats the inner posterior surface of the eyeball. Interestingly, the retina of vertebrates is inverted in the sense that the photosensitive segments of the receptor neurons are facing the outermost region of the eye. Because the retina is in the posterior part of the eye, the light beam arriving frontally through the lens must pass through the entire retinal thickness to stimulate the receptor cells. Its structure is organized into 3 layers where neuronal bodies are found. These layers are separated by 2 interfaces, where the synaptic connections between different retinal neurons are observed. The receptor cells are located adjacent to the ocular pigment epithelial layer, which is rich in melanin, allowing it to absorb reflections of light, preventing them from returning to the photoreceptors and blurring the image. Furthermore, the retinal pigment epithelial layer provides a continuous flow of retinal vitamin A to the photoreceptors where its fixation to the opsin molecule occurs. There are 2 types of receptor cells which are sensitive to light, rods and cones which convert light energy to electrical activity. The cones are responsible for daytime vision and distinction of colors and brightness. The rods, in turn, are responsible for vision in dim light and the distinction of light and dark. Most mammals have cones which are sensitive to wavelengths of light corresponding to the visible green and red colors. Primates, in turn, have 3 types of cones: those sensitive to the range of light wave, which we decode as red, those sensitive to green, and those that respond to blue. 15. This particular sensitivity of the cones is due to its photoreceptors, i.e. proteins called photopsins. There are 3 types of photopsins, whose maximum response corresponds to 3 ranges of light wavelengths. According to its photopsin, there are cones, which are sensitive to short wavelengths of about 420 nanometers, the S cones, blue. The cones, which are sensitive to waves of medium length, in the range of 530 nm, are the M cones, green, and cones sensitive to long. 14 Helga Kolb. Gross Anatomy of the Eye. In, Kolb H., Fernandez E., Nelson R., editors. Web Vision, the organization of the retina and visual system. Last updated the 25th of January, 2012. Available from http://webvision.med. Utah. edu/book/part I Foundations/Gross Anatomy of the A/. 15 Helga Kolb. Photoreceptors. In, Kolb H., Fernandez E., Nelson R., editors. Web Vision, the organization of the retina and visual system. Last updated the 25th of January, 2012. Available from http://webvision. Med. Utah. edu/book/part 2 Anatomy and Physiology of the Retina /photoreceptors/. 5. Waves are the L cones, red, which respond to the range of approximately 560 nm. 16. The gradient sensitivity of the cones together allows the detection of a wavelength range of about 380 to 780 nm, i.e. visible light, if red and green receptors are equally stimulated, we see yellow, and if the three types of cones are stimulated homogeneously, we see white. 17. The three cones, acting together, distinguish 200 shades, 20 saturation levels and 500 levels of brightness. The combination of these parameters provides 2 million gradations of colors. 18. The second types of photosensitive cells, rods, also have a light reactive pigment, rhodopsin, formed from a precursor and vitamin A. The degradation of rhodopsin by light energy causes excitation and response of the rods, which are responsible for seeing white, black, and bright/dark. The receptor cells are connected to other neurons, called bipala, which in turn make connection to the ganglion neurons, which conduct stimulation out of the retina. The signal emitted by various cones is assembled by horizontal cells, which determine how many cones converge to a ganglion cell. Regarding the rods, this convergence role is played by the amecrine cells located near the retinal surface, near the ganglion cell layer. The cell bodies of the bipala and horizontal cells form the inner nuclear layer of the retina. This layer is separated from that of photoreceptor cells by the outer plexiform layer, where the synapses among neurons of the two layers are located. The inner nuclear layer is separated from the ganglion cells layer by the inner plexiform layer, where the synaptic connections occur. The axons gathered from the ganglion neurons form the optic nerve. The stimulus is then driven back to the front by these cells, which merge to the optic nerve. The electrical impulses generated are organized and transmitted to the brain by the optic nerve. 19. When the fibers that make up the left and right optic nerves reach the base of the diencephalon, in its path toward the posterior area of the brain, they partially cross to the opposite side. In humans, in the structure called optic chiasm, 60% of fibers cross to the contralateral hemisphere, decussation, while the others continue their way. 16 James K. Bowmaker and Herbert J. Dartnell. Visual pigments of rods and cones in a human retina. J. Physiol. 1980, 298:501-11. 17 Helga Kolb. How the Retina Works. MSCI. 2003, 91:2835. 18 Titus Villies. The Physiology of the Senses. Lecture 1, The Eye. In The Physiology of the Senses. Transformations for Perception and Action, online course, revised 16 August 2012. Available from http://www. Tutis. ca/senses/l1i/l1i. PDF. 19 Dale-Pavsit al. Editors. Vision, the eye. In Neuroscience. 3 NDED. Sunderland, Ma, Seenauer. Associates, 2004. p.229-58. Available from http://www. CNS. Disc. Earn it. In /NS201_13/books/neuroscience_PVS. PDF. 6. Ipsilaterally. After the optic chiasm, the axons of ganglion cells begin to form the optic tract, which contains fibers from both eyes. This complex architecture makes the images originating from our right visual field be processed also in the left hemisphere of the brain and vice versa. The optic tract reaches the dorsal lateral geniculate nucleus. The neurons of the nucleus, in turn, through a portion of the so-called internal capsule. Optical radiation reach the primary visual cortex, known as Brodmann Area 17, or V1 in the calcarine fissure of the temporal lobe. In this area, the stimulus generated by the light on the retina will be decoded, interpreted and integrated, converting sensation to perception and cognitive phenomenon. This retinogeniculostriate route is the primary visual pathway 20. As seen above, in primates, color vision is the result of a complex integration of stimuli received by the three types of cones. This type of vision in normal individuals is called trichromacy. The absence or non-operation of one or more cone leads to different forms of color blindness, dyschromatopsia. About 8% of men and 0.5% of women have congenital abnormalities of color vision. 21. It is known that specific genes determine the presence of each type of cones. The genes encoding the red and green pigments are located in adjacent regions of the chromosome X22, which explains the differences in prevalence of congenital dyschromatopsias between men and women. Conversely, the gene for the blue pigment is at chromosome 7. 2. Olfaction. The sensory systems associated with the nose and the mouth are responsible for detecting chemicals in the environment. Thus, olfaction, taste and trigeminal tactile sensations are called chemical senses. The olfaction is important in the selection of food and for reproductive and maternal functions. The neuroendocrine regulation and emotional responses also undergo the influence of olfactory stimuli. 20 Dale-Pavsit al. Editors. Central Visual Pathways. In Neuroscience. 3. NDED. Sunderland, Ma. Seenauer Associates, 2004. p.259-82. Available from http://www. CNS. Disc. Earn it. In /NS201_13/books/neuroscience_PVS. PDF. 21 Michael Kalonatis and Charles Law. Color Perception. In Kolbach, Fernandez E., Nelson R., Editors. Web Vision, The Organization of the Retina and Visual System. Copyright 2014. Available from http://webvision.med. Utah. edu/book/part8 receptors/color perception/ 22 Jeremy Nathans et al. Molecular Genetics of Inherited Variation in Human Color Vision. Science. 1986, 232:203-10. 7. Olfaction allows one to identify a large number and variety of chemical substances from the external environment. It is estimated that human beings can sense about 10.000 to 100.000 chemical substances with different odors. 23. These odorants, unlike sapid substances, should be small and volatile molecules that can be transported by air from the emission source to the olfactory system. This system, which is one of the oldest in the phylogeny of mammals, is characterized by exceptional sensitivity and high discriminatory power. This last quality is verified by the fact that minor changes in the molecular structure of an odorant may determine perceptions of different odors. Transported by the inspired air, odorants reach the nostrils and nasal cavity. In this region, bone structures known as shells or turbinates direct the airflow to the rear and top of the cavity. In this region, a small area of the lining epithelium of the nasal cavity contains the endings of the olfactory sensory neurons. In humans, this area, i.e., the olfactory plaque, has 2 to 4 cm 2 and contains 10 to 20 million receptor cells. When the odorants reach the olfactory plate, they are dissolved in the mucus that covers the epithelium. In this mucus, odorant-binding proteins 24 and some specific antibodies provide transport and presentation of the odorant to the sensory endings of the receptor neurons. The cell membrane of these structures houses the so-called G proteins coupled receptors 25. These proteins are encoded by a large multigeny family 26. It is known that humans have about 330 odorant receptors 27 while mice have approximately 1028. These data suggest that between 1 and 2% of the human genome are genes dedicated to the production of these proteins. This number is second only to the number of genes related to the immune system. Each sensory neuron has only one type of odorant receptor 29, however each receptor can be coupled to various odorants 23. 23. Linda B. Buck. Unraveling the Sense of Smell. Nobel Lecture, 8 December, 2004. p.267-83. Cited 2013. December. Available from http://www. Nobel Prize. org/nobel_prizes/medicine/laureates/2004/Buck Lecture. PDF. 24. Paolo Pelozzi. Odorant Binding Proteins. Crid Rev Biochem Mol Biol. 1994, 29:199-228. 25. Peter Mombayes. 7 Transmembrane Proteins as Odorant and Chemosensory Receptors. Science. 1999, 286:707-11. 26. Linda Buck and Richard Axel. A novel multigeny family may encode odorant receptors, a molecular basis for odor recognition. Cell. 1991, 65:175-87. 27. Bettina Malnick et al. The Human Olfactory Receptor Gene Family. Procnate Lackard 2004, 101:2584-9. 28. Paula Godfrey, Bettina Malnick and Linda B. Buck. the mouse olfactory receptor gene. Family. Procnate Lackage SA 2004, 101:2156-61. 29 Bettina Malnickit al. Combinatorial receptor codes for odors. Cell. 1999, 96:713-23. 8. 2.1. Transduction. Once the coupling of the receptor molecule with the odorant is performed, the transduction process begins. This pathway in mammals is composed of a variable component, the olfactory receptor, and 4 constant elements, G-alpha olf heterotrimeric G protein, adenyl cyclase, which produces the second messenger, the cyclic AMP, CAMP, a channel for cations controlled by cyclic nucleotide, and a chlorine channel. Thus, the response of the sensitive olfactory neurons to different combinations of odorants results predominantly or solely from olfactory receptor 30. Different odorants are discriminated by combinatorial codes generated by the stimulation of different receptors yielding different olfactory perceptions. 31. 2.2. Communication among olfactory sensory cells. The sensory neurons cross the cribriform plate of the ethmoid bone, forming the olfactory nerve, and transmit the stimulus directly to a brain structure called the olfactory bulb, where they connect, in structures called glomeruli, with the second neuron in the chain, mitral cells or tufted cells. Each glomerulus contains apical dendrites of approximately 25 mitral cells, which connect with the axons of about 25,000 olfactory receptor neurons. This convergence probably increases the signal and the sensitivity of mitral cells. In the glomeruli, dendrites of about 50 tufted cells and 25 periglomerular cells are also observed. The function of the latter is not well known, yet it is assumed that they exert control functions. 32. The innermost layer of the olfactory bulbs of vertebrates consists of granular cells, which establish synapses with the basal dendrites of mitral cells. These connections are dendrodendritic since these cells have no axons. Its probable role is establishing lateral inhibitory circuits and acting on the plasticity of the olfactory bulb. Granular cells and periglomerular are some of the few neurons that are renewed throughout life. 33. 30 Peter Mombayes. Genes and ligands for odorant, vomeronasal and taste receptors. Nat Rev Neurosci. 2004, 5263-78. 31 Kentaro Kajia et al. Molecular bases of odor discrimination, reconstitution of olfactory receptors that recognize overlapping sets of odorants. J. Neurosci. 2001, 21:6018-25. 32 Denise de Arruda. The periglomerular cell of the olfactory bulb and its role in the odor processing, a computational model, thesis. Ribeirão Preto, SP, Faculdade de Filosofia, Ciências e Letras de Ribeirão Preto, 2010. 33 Dale Pavsit al. Editors. The Chemical Senses. In Neuroscience. 3. NDED. Sunderland, Ma. C. Nauer Associates, 2004. p.337-67. 9. 2.3. Integration in the central nervous system. The axons of the mitral cells together form the lateral olfactory tract. Through it, The olfactory bulb sends projections to structures that, as a group, are called primary olfactory cortex. These structures can be divided into three groups: the anterior olfactory nucleus, the rostral olfactory cortex, comprising indusium gryzeum, the anterior continuation of the hippocampus, the tenure tector, infralimbic cortex, and the lateral olfactory cortex that includes the piriform, perimygdaloid, transitional and anterinal cortices. The primary olfactory cortex sends projections to cortical and subcortical brain areas that are not considered as part of the olfactory system. Thus, the primary olfactory cortex and anterior olfactory nucleus sends efferent fibers to the thalamus and hypothalamus and some neurons in piriform cortex innervator region of the orbitofrontal cortex, revealing multimodal neurons that respond to olfactory and gustatory stimuli. Along this path, odorants affect cognitive, visceral, emotional and homeostatic behaviors. 34,35. 3. Gustation. The words taste and flavor are often used indiscriminately. However, taste is one of our senses, while flavor is a more complex perception, composed of taste, olfactory, and tactile sensations. The sense of taste, or gustation, allows us to perceive and distinguish the presence of sapid substances in food and whatever is brought into the mouth, thus allowing one to ingest nutrients selectively and avoid ingesting poisons and toxins. It is accepted that humans can distinguish 5 different tastes: sweet, salty, bitter, acid or sour, and umami. The latter, recently recognized, is the sensation caused by monosodium glutamate as well as the amino acid L-aspartate 36. The existence of a taste sensation associated specifically to fat is debatable. 37. The gustatory pathway begins with the gustatory sensory cells. There are 4 types of such cells, where type I have the function of support, while type 4 probably. 34. Michael T. Shipley and Matthew Ennis. Functional Organization of Olfactory System. J. Neurobiol. 1996, 30:123-76. 35. Dale Pavsit al. Editors. The Chemical Senses.in. Neuroscience. 3. NDED. Sunderland, Ma. Seenauer Associates, 2004. p.337-67. 36. Bernd Lindemann et al. The Discovery of Umami. Chemistry Senses. 2002, 27:843-4. 37. Richard D. Meertz. Is there a fatty acid taste? A new Rev Neuter. 2009, 29:305-27. 10. Generates the other types. Both lack the capacity to generate stimuli decodable as taste. Sensation. This function is performed by types 2 and 3. 38. Taste sensory cells gather into taste buds. In these structures, 50 to 100 taste cells are clustered in an oval shape. A small opening, the taste pore, situated at the apex of the bud allows the entry of sapid substances. The taste buds are located in the so-called gustatory papillae, which are visible on the surface of the tongue. There are 4 types of papillae. The filiform papillae are the most numerous. However, they have no taste sensivity. Its function is tactile sensitivity. The fungiform Papillae are mushroom-shaped and cover the anterior 2/3 of the tongue and are innervated by the chorda tympani, one of the branches of the intermediate facial nerve. There are about 200 of these papillae on the tongue, containing 25% of the taste buds. It is estimated that the tongue has about 1,250 taste buds in fungiform papillae. The foliate papillae are located on the sides of the tongue, and are more sensitive to acid taste. These papillae are innervated by the glossopharyngeal nerve. On average, 2 of those buds occur at the posterolateral tongue with approximately 600 taste buds each, or approximately 25% of taste buds of the tongue. The circumvallate papillae are the largest ones, with a peculiar shape. They are elevations with a central depression, surrounded by a high wall. They are located on the back of the tongue, forming the so-called V lingual. They are sensitive to sour taste, and particularly to bitter taste. Their number varies from 3 to 13, containing 25 taste buds each. Therefore, these buds harbor about 50% of the tongue taste buds. The nerve responsible for the transmission of stimuli received by them is the glossopharyngeal 39. There are 2,500 other taste buds scattered in the oropharynx, proximal esophagus, and epiglottis. Its innervation is made by the vagus nerve. 3.1 Transduction. The sapid molecules, which are responsible for these 5 tastes, are dissolved in saliva and penetrate the taste buds through its pores and come into contact with receptors on microvilli of type 2 taste cells. Then they bind to receptors termed GPCR. G-proteins coupled receptor for sapid substances. G-proteins are a class of proteins, of multigenic origin, present in mammals, which bind to sapid substances or odorants. 38 Nirupar Chodhari and Stephen D. Roper. The Cell Biology of Taste. J. Cell Biol. 2010, 190:285-96. 39 Dale Pavsit al. Editors. The Chemical Senses. In Neuroscience. 3. NDED. Sunderland, Ma. C. Nauer Associates, 2004. p. 337-67. 11. From the activation of these receptors or ionic channels, the process of transduction is started, i.e. the cascade of reactions that ultimately promote membrane depolarization of the taste sensory cells, converting chemical stimulus into electrical impulses carried along nerve pathways to the central nervous system, where integration and decoding of these impulses occur. An important family of these receptors is composed of members called T1R1, T1R240, and T1R3, which have been more recently discovered 41. This family of GPCRs mediates the pleasant, sweet and umami tastes. These molecules are associated in complex homodimeric or heterodimeric receptors, forming molecules that bind to substances with a sweet taste, whether or not saccharides. T1R receptors are critical for the detection of sweet tastes and are expressed in subsets of sapid substances. Receptor cells TRC. There are 3 groups of such cells: the co-expressing T1R1 and T1R3, T1R1 plus 3, the co-expressing T1R2 and T1R3, T1R2 plus 3, and those expressing only T1R3. It has been shown that the T1R2 plus 3 heterodimer is the sweetness receptor. that responds to all categories of sweet substances, whether natural sugars, artificial sweeteners and D-amino acids. 42. The umami taste is given by binding monosodium glutamate to receptors such as taste mGLUR4, taste mGLUR1 or to the dimer T1R1/T1R3, T1R1+3, resulting from the combination of the two T1 receptors. 43,44. The GPCRs for bitter taste belong to another large family called T2R45. After the stimulation of GPCR, transduction of sweet, bitter and umami tastes occurs through two main pathways, mediated by molecules that act as second messenger in this cascade. One of them is the cyclic adenosine monophosphate, CAMP. In the case of the gustatory path, a specific G protein, known as G-gusteucin, has been described and is found primarily in cells expressing T2R46, whose activation correlates inversely with the concentration of CAMP. The changes in CAMP concentration finally leads to other phosphorylation reactions, which cause inhibition of the potassium channels of the taste sensory cells, which depolarizes the cell and causes 40 marker unit al. Putative mammalian taste receptors, a class of taste-specific GPCRs with distinct topographic selectivity. Cell 1999, 96:541-51. 41 Mariana Maxit al. TARS1R3, encoding a new candidate taste receptor, is allelic to the sweet responsiveness locus SAC. Nat Genet 2001, 28:58-63. 42 Greg Nelson et al. Mammalian sweet taste receptors. Cell 2001, 106:381-90. 43 Greg Nelson et al. An amino acid taste receptor. Nature 2002, 416:199-202. 44 Nirupar Chodhari. Taste receptors for umami, the case for multiple receptors. J. Klin-Neuter. 2009, 90:738s742s. 45 Elliott Adler et al. A novel family of mammalian taste receptors. Cell 2000, 100:693-702. 46 Susan K. McLaughlin et al. Gusteucin is a taste cell-specific G protein closely related to the transducins. Nature 1992, 357:563-9. 12. The secretion of neurotransmitters. Another second messenger is the release of intracellular calcium mediated by inositol triphosphate, IP3, with activation of the melastatin-type transient receptor potential cation channel TRPM5, depolarizing the cell and releasing the ATP neurotransmitter 47. The activation of TRPM5 may be the common pathway in the transduction of sweet, bitter and umami tastes. 48. Sour tasting substances can stimulate specific channels, PKD2L1, or act through the epithelial sodium channels. Sour taste is perceived in type 3 sensory cells. The salty taste is transduced by the entry of sodium in the cell through sodium channels, called NACs, similar to those existing in the kidneys. The rise of the sodium concentration in the cell leads to depolarization and generation of neurotransmitters. 49. 3.2. Communication among gustatory sensory cells. Type 2 gustatory cells are just receptors because they do not establish bonds with neurons that run to the central nervous system. Type 3 cells, in turn, communicate directly with nerve fibers by means of synapses and send signals to the central nervous system. Consequently, the continuity of transmission of stimulus requires communication between type 2 and 3 cells. When stimulated, the latter release neurotransmitters, probably serotonin, to the sensory nerve fiber, which depolarizes and transmits the impulse to the brain. 3.3 Integration in the central nervous system. All stimuli caused by sapid substances trigger reactions of different qualities and intensities in the sensory cells, which are picked up by the facial, glossopharyngeal and vagus nerves and carried to the spinal cord and the nucleus of the solitary tract, which triggers non-conscious reflexes of rejection or acceptance of stimuli. Based on that, new nerve connections transmit stimulation to the so-called primary gustatory cortex, within the operculum-insular area. Neurons in this area, in monkeys, respond to the identity and intensity of flavor. Its response is not attenuated by satiety. New connections lead to 47 Robert F. Margolsky. Molecular mechanisms of bitter and sweet taste transduction. J. Biochemistry. 2002, 277:1-4. 48 Yu Feng Zhang et al. Coding of sweet, bitter, and umami tastes, different receptor cells sharing similar signaling pathways. Cell. 2003, 112:293-301. 49 Jairam Chandrasekhar et al. The receptors and cells for mammalian taste. Nature. 2006, 444:288-94. 50 Eugene Huang et al. Presynaptic, type 3, cells in mouse taste buds sense sour, acid, taste. J. Physiol. 2008, 586:2903-12. 13. The integrated stimulus into the secondary gustatory or orbitofrontal cortex. This area represents sensory, affective and hedonic food qualities. This is where the integration with the olfactory, visual and tactile sensations occurs. The ingestion control is in this area, which is also integrated into the system of reward and punishment. 51. The perception of taste can be produced from two models of neural representation of stimuli received by sensory cells. The so-called labeled line hypothesis states that a particular taste quality activates certain neurons, which otherwise would remain quiescent in the absence of the stimulus. The across neuron pattern hypothesis states that there are no specific neurons for the tastes that are determined by a matrix activation of multiple neurons corresponding to a pattern that correlates with a given taste. Current knowledge seems to confirm the labeled line hypothesis. 52. The gustatory neural code also seems to involve a temporal aspect. 53. 4 chemosensory trigeminal system. Such a system consists mostly of multimodal nociceptive receptor neurons, whose axons run along the branches of the trigeminal nerve. These nociceptor neurons are typically stimulated by chemicals which are considered irritant, such as sulfur dioxide, ethanol, acetic acid, ammonia, menthol and carbon dioxide, in addition to capsaicin. Its function is to alert the organism about the potential risk of chemicals that can be ingested, inspired, or have contact with the eyes or other facial structures. This information is carried by the ophthalmic, maxillary and mandibular branches to the spinal component of the trigeminal ganglion, which sends them to the ventral posterior lateral nucleus of the thalamus and then to the somatosensory cortex, which processes the irritation and oral and facial pain. Some of the mouth nociceptive neurons send their axons to the branches of the vague and glossopharyngeal nerves. In addition to the nociceptive neurons, the oral mucosa houses mechanoreceptors whose afferent fibers run by infraorbital nerves, chorda tympani, lingual and glossopharyngeal. These receptors allow the distinction of textures 54,55. 51 Edmund T. Rolls and Leslie L. Bailey's gustatory, olfactory, and visual convergence within the primate orbitofrontal cortex. J. Neurosci. 1994, 14:5437-52. 52 Jayaram Chandrasekhar et al. The receptors and cells for mammalian taste. Nature. 2006, 444:288-94. 53 Lemon Ch and Katz DB. The Neural Processing of Taste. BMC Neurosci. 2007, 8, Supl 3s5. 54 Dale Pavsit al. Editors. The Chemical Senses. In Neuroscience. 3. NDED. Sunderland, Ma. C. Nauer Associates, 2004. p.337-67. Cited 2013 December. http://www. CNS. Disc. Earnit. in/201_13/books/neuroscience_pvs. PDF. 55 Martha R. Bajek and Gary J. Pickering. Astringency, Mechanisms and Perception. Crid Rev Food. SCI Neuter. 2008, 48:858-75. 14. Chemical Origin of Wine Attributes. 1. Color. A plain visual inspection allows one to establish the type of wine, and predict, despite not so precisely, some of its features. The origin of the color of the wine is due to pigments, primarily originating from the grapes, but also from their contact with wood. Certain wavelengths of the incident light spectrum on wine are absorbed by the pigments and others are reflected. Thus, the color of red wine results from the absorption of all wavelengths except the red one. The pigments, which are responsible for the wine color, are the anthocyanins that are present in grapes in the form of glucosides, compounds of glucose and a flavonoid component called anthocyanidin. Glucose may also bind to organic acids such as caffeic, coumaric and acetic. The position of the methyl and hydroxyl groups that bind to the B ring of anthocyanidin determine 5 different types of anthocyanins: cyanin, delphinidin, malvidin, ponin and petunidin. The bluish hues increase with the number of free hydroxyls while the red hue increases with methylation. Malvidin, which is the most abundant anthocyanin in grapes and the most methylated, contributes a great deal to the intense color of young red wines. 56. Moreover, each of anthocyanins in wine occurs in a 5-state molecular dynamic balance. one of them is linked to sulfur dioxide. The remaining ones, within the pH range of the wine, tend to be colorless. A small proportion, which exists in the form of the flavolium cation, is responsible for the red color. This ratio is pH dependent, increasing with its decrease. The rise in pH causes increased quinoidal state of anthocyanins, changing the color of the wine to the blue-violet 57. The aging of red wine brings, as a consequence, a well-known color change, which shifts the purplish tone gradually to red, and subsequently, to orange and yellow hues. A major cause of this phenomenon is the formation of so-called polymeric pigments, which are less sensitive to pH and carbon dioxide, allowing the expression of their color. An important compound is the polymer formed by the anthocyanins with 56 Ronald S. Jackson. Chemical constituents of grapes and wine. In Wine Science, Principles, Practice, Perception. 2. NDED. Burlington, Ma, Academic Press, 2000. p. 232-75. 57 Yam Gallet and James Crum, editors. Phenolic compounds. In Concepts in Wine Chemistry. 2nd ed. South San Francisco, CA, The Wine Appreciation Guild, 2004. p. 95-129. 15. Procyanidins. Other important pigments are pyranoanthocyanidins, which are more intensely orange-colored than the anthocyanins, which accounts for 50% of the pigments present in a 5-year-old wine. 58. The elements responsible for the color in white wine are less known, but it is believed that the light straw color, often displayed by these wines, is conferred by a limited extraction and oxidation of flavonols such as quercetin and kaempferol. The golden color acquired with age is derived from the oxidation of phenols and galactronic acid, as well as the caramelization of sugars by Maillard reactions. 59. 2. Aroma. Aromas present in wines are traditionally classified as primary, secondary or tertiary. Primary aromas derive from substances contained in grapes, which remain more or less time in the wine. Secondary aromas are created by fermentation processes and wood aging. Tertiary aromas, in turn, are those that develop over the years, for wines with aging potential in the bottle and in the absence of oxygen, reductive aging. This combination of aromas is called bouquet. Likewise, defective aromas occur, which must be identified by a taster and may disqualify the wine for consumption. 2.1. Primary aromas. Grapes from the vast majority of cultivars Vitis vinifera do not exhibit characteristic aromas. Some exceptions are the floral or citrus scent present in Muscat varieties, vegetable or bell pepper flavors, observable in Cabernet Sauvignon grapes and related cultivars, such as Merlot, Cabernet Franc, Petit Verdot and Carmenère. In the case of Muscat family, Muscat of Alexandria, Muscat Canely, Muscat Hamburg, Muscat Orange and Muscat Ottonelle, and other related varieties, Riesling, Sylvaner, Jewers Traminer, Muller Thurgau and others, the primary aroma is due to the presence of grape compounds known as monoterpenes, derived from the basic structure isoprene. 58 Celestino Santos Puelga and Victor de Freitas. Influence of phenolics on wine organoleptic properties. In Moreno Arribas MV, Polo MC, editors. Wine Chemistry and Biochemistry. New York. Springer, 2009. 59 Ronald S. Jackson. Chemical Constituents of Grapes and Wine. In Wine Science, Principles. Practice, Perception. 2. NDED. Burlington, Ma, Academic Press, 2000. p. 232-75. 16. 2-methyl-1,3-butadiene, C5H8. Monoterpenes are usually hydrocarbons, myrcene, limon, but they also exist as alcohols, geraniol, nerol, linalool, and aldehydes. Geraniol, nerol, 60. Whenever a muscat wine exhibits lemon scent, this is because a terpene, responsible for the lemon aroma of the lemon itself, was also present at that grape from where the wine was originated. In the case of Cabernet Sauvignon and associated grapes, vegetable or bell pepper aroma is due to the presence of pyrasic compounds in the fruits, especially 2-methoxy-3-isobutylpyrazine. The concentration of this compound is higher in grapes from cooler regions and especially in those that are not harvested at full maturity. 60. Another cultivar, which is rich in pyrazines, is the Sauvignon Blanc. 61. Wine grapes contain numerous non-volatile compounds and therefore non-odorants, which are precursors of wine aromatic components. They are phenolic acids, cysteine S conjugates, carotenoids, unsaturated lipids, S-methylmethionine and glycoconjugates. Phenolic acids are hydroxycinnamic acids, which occur in grapes as esters of tartaric acid. The most common are caphteric acid and cuttaric acid, which give rise to the odorants vinyl derivatives 4-vinyl phenol and 4-vinyl guaracol present in wine. This reaction is mediated by the cinnamate decarboxylase of Saccharomyces cerevisiae. Catechins inhibit this conversion and the aromas attributed to these phenols are not too significant in red wines. However, catechins do not inhibit cinnamate decarboxylase of Brettanomyces contaminating yeasts and there may be a transformation of vinyl phenols into ethyl phenol, generating barnyard, stallion, and mouse defective aromas. In white and rose wines, aromatic vinyl phenols can exert some influence. Vinyl guaiacol can contribute to the varietal flavor of Juer's Traminer. 62. One of the few phenolic derivatives, presenting as a primary component of grapes, is methylanthranilate ester. This substance is present in significant amounts in grapes from Vitis labrusca cultivar and is responsible for the characteristic aroma of 60. Jan Margalit and James Crum, editors. Aroma and Flavor. In Concepts in Wine Chemistry. 2. N.D. ed. South San Francisco, CA, The Wine Appreciation Guild, 2004. p. 133-92. 61 Ronald S. Jackson. Olfactory System. In Wine Tasting, A Professional Handbook. London. Academic Press, 2002. p. 39-78. 62 Raymond Baums. Wine Aroma Precursors. In Moreno Arribas MV, Polo MC, editors. Wine Chemistry and Biochemistry. New York, Springer, 2009. p. 251-74. 17. Wines which are made with these varieties, foxy methylanthranilate can be detected in small quantities in wine grapes such as Pinot Noir, Riesling and Sylvaner. 63. S-cysteine conjugates are non-volatile precursors and non-odorants of volatile and odoriferous thiols. These compounds are derived from the L-cysteine molecule with a carbon chain linked to the sulfur atom. The activity of an enzyme, the cysteine S conjugate lyase, during alcoholic fermentation by Saccharomyces cerevisiae breaks the thioester bond, releasing the aromatic thiol fraction. Thiols are also called mercaptans, and the most important one in wine are 4-mercapto-4-methylpentan 2,1,4-MMP, 3-mercaptohexane-1-ol acetate, a 3-MH, 3-mercaptohexane-1-ol, 3-MH, 4-mercapto-4-methylpentan 2-ol, 4-MPO, and 3-mercapto-3-methylbutan-1-ol. 3-MMB, 4-MMP gives Sauvignon Blanc wines a strong odor of boxwood and broom shrub. 3-MH is associated with the aroma of grapefruit. This latter compound and its derivative, a 3-MH, also make up the aromatic profile of the passion fruit. 3-MMB, on the other hand, has an aroma of cooked leeks 64,65. Grape carotenoids, represented primarily by carotene, are precursors of C13. Norisoprenoids, which are potent odorants. The paths of formation of these compounds, such as alpha and ianone, are not well known. The latter gives a scent of violet and raspberry to the wine 66. Grapes contain unsaturated fatty acids such as linoleic and linolenic acid. These lipids are released after the crushing of the grapes in the pre-fermentation stage, and are degraded by the lipoxygenase enzyme in C6 compounds such as hexanol and 2-hexenol, which have vegetable, grassy aromas 66. The S-methylmethionine, SMM, originated from grapes, is also present in wine. After bottling and proper storage, under anaerobic conditions, the SMM is converted to dimethyl sulfide. This compound is a powerful odorant and, depending on its concentration, it can bring fine or defective aromas to the wine. In small quantities, the characteristic aroma is corn or asparagus. At higher concentrations, the odor shifts to cooked cabbage or shrimp. At concentrations present in high-quality wines, subject to 63 Ronald S. Jackson. Chemical constituents of grapes and wine. In Wine Science, Principles, Practice, Perception. 2. NDED. Burlington, Ma, Academic Press, 2000. p.232-75. 64 Philippe Dariet et al. Un paradoxu, les composes soufres volatiles responsables de defaults et de qualités dans les vins. J. int sci vin vin. 1999, 33, Spec 137-43. 65 Dennis Dubadieu and Takato Shitaminaga. Polyfunctional thiol compounds. In Moreno Aribas MV. Polo MC, Editors. Wine Chemistry and Biochemistry. New York, Springer, 2009. p.275-93. 66 Raymond Baums. Wine Aroma Precursors. In Moreno Aribas MV, Polo MC, Editors. Wine Chemistry and Biochemistry. New York, Springer, 2009. p.251-74. 18. Long storage. This compound is related to the fine truffle aroma that these wines may present 66. Some volatile substances are present in grapes from Vitis vinifera linked to Glycosides, forming glycoconjugates. There are different subtypes of these compounds. According to the glycidic fraction, APOs, ramous and arabinos. These groups may be bound to monoterpenes, alcohols, phenols, volatile N-isoprenoids, among others. These odorous compounds are progressively hydrolyzed by glucosidase 67. 2.2. Secondary aromas. Secondary aromas are generated by fermentation processes and wood aging. Fermentation adds little to the aroma of wine. This reaction is exergonic, releasing energy as heat, which along with plenty of fermentable sugars and oxygen availability, provides a huge proliferation of yeasts and the formation of many other substances other than ethanol by its metabolism. Some of these substances are neutral or volatile. Esters 68. There are about 300 of these molecules in wine and some with aromatic importance. Acetates are formed by a reaction between ethanol and other short-chain alcohols, isomal alcohol and isobutyl alcohol, which are formed, and acetic acid. Due to their low molecular weight, these esters are volatile and strongly influence the aroma of young wines, contributing to its fruity character. They are therefore called fruity esters. Isomal acetate has a strong odor of bananas, often recognized in Beaujolais, Nouveau and other carbonic maceration wines. Benzyl acetate has an aroma similar to apples. In turn, ethyl acetate, which presents an odor of nail polish or model airplane glue, may be present in botrytized wines, since the microperforations caused by Botrytis cinerea in the grape exocarp allow penetration of acetic bacteria. This organism converts ethanol into ethanoic acid, acetic acid, which generates ethyl acetate 69. Apparently, acetates bring a greater contribution for aroma in wine than ethyl esters of fatty acids such as ethyl hexanoate, ethyl octanoate and ethyl decanoate. However, the 67 Raymond Baum's Wine Aroma Precursors in Moreno Arribas MV, Polo MC, Editors. Wine Chemistry and Biochemistry. New York, Springer, 2009. p.251-74. 68 Yamar Gallet and James Crum, Editors. Must and Wine Composition. In Concepts in Wine Chemistry. 2. NDED. South San Francisco, CA, The Wine Appreciation Guild, 2004. p.1-55. 69 Ronald S. Jackson. Chemical Constituents of Grapes and Wine. In Wine Science, Principles. Practice, Perception. 2. NDED. Burlington, Ma, Academic Press, 2000. p.232-75. 19. Combination of these two ester groups is responsible for a higher aromatic wine quality 70. Another class of substances responsible for secondary aromas are small molecule organic acids, the so-called short-chain fatty acids. Due to its low molecular weight, these compounds volatilize easily from the wine, constituting the so-called volatile acidity. The metabolism of yeast is responsible for the synthesis of several of these fatty acids. The most important among them is acetic acid, which, at concentrations below recognition thresholds, about 400 mg/L, increases wine complexity. Above this limit, a vinegar odor becomes recognized, which is considered a defect. Acetic acid accounts for over 90% of volatile acidity. Other acids of this class are formic acid of pungent odor, propionic acid with an odor of fat, and butyric acid with an odor of rancid butter. Acids with longer chains, between 6 and 10 carbons, have an odor which is described as goat odor. 71. Wood staging transfers substances to the wines, which are responsible for other secondary aromas. Among these, methyl-octalactone is the source of the scent of coconut, whereas vanillin is responsible for the aroma of vanilla, and benzaldehyde, for aroma of bitter almonds. 72,73. 2.3. Tertiary aromas. Tertiary aromas are those that develop over the years on wines with potential bottle aging, in the absence of oxygen, reductive aging. This combination of aromas is called bouquet. Its development is accompanied by the loss of varietal and fruity aromas. Therefore, long-term storage of wines must bring aromatic complexity and softness to compensate for this loss. The genesis of the bouquet is not well known. In aromatic white wines, the loss of terpenes and the appearance of their oxides is characteristic. These compounds have a higher threshold of perception and different aromatic characteristics. Thus, linalool oxide has a eucalyptus flavor instead of the floral scent of linalool. Other processes can generate tertiary aromas. One of them, which has been already discussed, is the hydrolysis of glycoconjugates, releasing 70 Ca van der Merwe, Cj van Wyk. The contribution of some fermentation products to the odor of dry white wines. J. Inol Vitek. 1981, 32:41-6. 71 Maurizio Ugliano and Paula Henschke. Yeasts and Wine Flavor. In Moreno Arribas MV, Polo MC. Editors. Wine Chemistry and Biochemistry. New York, Springer, 2009. p.313-92. 72 Ronald S. Jackson. Post-fermentation treatment and related topics. In Wine Science, Principles. Practice, Perception. 2. NDED. Burlington, Ma, Academic Press, 2000. p.355-433. 73 Yanmar Gallet and James Crum, Editors. Oak Products, Cooperage and Cork. In Concepts in Wine Chemistry. 2. NDED. South San Francisco, CA, The Wine Appreciation Guild, 2004. p.217-63. 20. Aromatic substances. With aging, the hydrolysis of these compounds may release aromatic molecules, such as volatile phenols. At the same time, the degradation of wine carbohydrates gives rise to highly aromatic compounds, such as the aldehyde 2-furfuryl, with its caramel odor. As described above, another molecule which apparently has an important role in the bouquet is dimethyl sulfide, which is formed from precursors such as amino acids containing sulfur. The formation of the reduction bouquet is also characterized by a decrease in the concentration of acetate and ethyl esters, with the exception of increasing concentrations of ethyl esters from diprotic acids. 74,75. 2.4. Aromatic defects. 2.4.1. Oxidation. The main cause for oxidation is an inadequate storage of wine, when bottles are left standing or under excessive heat. Such conditions affect the characteristics of the cork, allowing air to enter the bottle and the contact of the components of wine with oxygen. The reaction of oxygen with ethanol produces acetaldehyde, a substance of strong aromatic intensity, which resembles the scent of oloroso sherry, toasted walnuts, or bruised apples. 76. 2.4.2 Reduction. Modern winemaking practices often tend to process wines in a reductive environment, systematically isolating it from oxygen. This practice is not innocuous, as it may cause some defects in wine, such as the reduction odors, also called odors of bottle or of light or of sun. The photochemical effect of light enhances this problem. The reduction odor is described as a lack of aromatic sharpness, slightly unpleasant garlicky odor, which may resemble sweat. A reductive medium promotes the formation of sulfur compounds such as hydrogen sulfide, mercaptans, thiols and dimethyl sulfide. Defective aromas associated with these compounds are the smell of rotten eggs, cabbage, burned rubber and cooked corn. Other so-called varietal characteristics, such as the smell of cat urine and passion. 74 Perez Prieto L.J., Lopez Rica J.M. and Gomez Plaza E. Differences in major volatile compounds of red wines according to storage length and storage conditions. J. Food Comp Anal. 2003, 16:697-705. 75 Yamar Gallet and James Crum, editors. Oxidation and Wine Aging. In Concepts in Wine Chemistry. 2. NDED. South San Francisco, CA, The Wine Appreciation Guild, 2004. p.193-215. 76 Ronald S. Jackson. Sensory Perception and Wine Assessment. In Wine Science, Principles. Practice, Perception. 2. NDED. Burlington, Ma, Academic Press, 2000. p.544-90. 21. Fruit in Sauvignon Blanc wines are knowingly derived from 4-mercapto-4-methyl pentane 2-1 and 3-mercaptohexanol acetate, both volatile sulfur compounds. 77. Two factors contributing to the decrease in redox potential are the concentration of sulfur dioxide, SO2, and Lees, which act as antioxidants by absorbing oxygen. Another cause of aromatic defects associated with reduction may be the current trend of closing bottles with a screw cap system, which seals the container more effectively than caps made of cork and polymers. Some aromatic defects, particularly the smell of rubber, have been associated with this form of bottle seal. 78. The great art of the enologist is to balance reductive winemaking with small contributions of oxygen, so that the true varietal characteristics and terroir are expressed, in addition to preserving the freshness and fruitiness enabled by oxygen-free winemaking. Enological wisdom, which has developed for centuries, gives us a clear example of this balance. White wine resting on its lees, it has been said that dead yeast act as antioxidants, reducing redox potential, tend to develop reduction aromas. The centuries-old practice of battonage, in addition to extracting favorable products from the mass of yeast, introduced small amounts of oxygen into the wine, restoring the balance between reduction and oxidation. 2.4.3. Corky. The defect known as corky is characterized as a moldy, cardboard or wet dog odor. Different compounds can cause these defective odors in wine. The most important of these is 2,4,6-trichloronisole, 2,4,6-TCA, which is a product from the activity of fungi present in the cork. This substance, together with 2,3,4,6-tetrachloronisole and pentachloronisole, accounts for at least 80% of cases of corky wine. The prevalence of this defect in wines is 2 to 7%. The likely route for the synthesis of 2,4,6-TCA is the O-methylation of highly toxic chlorophenols precursors such as the fungicide pentachlorophenol. Chlorine from cork bleaching with hypochlorite may also be involved in the genesis of anisols. Although several strains of fungi convert 2,4,6-trichlorophenol to 2,4,6-TCA, the Trichoderma and Fusarium strains are the most. 77 Jan Margalit and James Crum, editors. Aroma and Flavor.in. Concepts in Wine Chemistry. 2. N.D. ed. South San Francisco, CA, The Wine Appreciation Guild, 2004. p.133-92. 78 Peter Godinit al. Towards offering wine to the consumer in optimal condition, the wine, the closures, and other packaging variables, a review of ARI research examining the changes that occur in wine after bottling. Wine in J 2005, 2:20-30. 22. Active. Other moldy odors can be attributed to the production of guaiacol, 2-methoxyphenol, by fungi of the genera Penicillium and Aspergillus. Other substances, such as geosmin, trans 1,10-dimethyl trans 9-decalol, and R1-octan-3-ol, originating from the fungal metabolism, can contribute to the musty odor of wines 79-80-81. 2.4.4. Acetic acid and ethyl acetate. The formation of acetic acid is due to the bacteria Acetobacter and Glucanobacter, which can proliferate in wine in the presence of oxygen. These microorganisms oxidize ethanol to acetaldehyde, which, in turn, is oxidized to acetic acid. As seen above, acetic acid accounts for over 90% of the volatile acidity of the wine, usually between 0.2 and 0.4 g/L. The concentration of acetic acid in wine must not exceed 0.7 g/L. Above this concentration, a vinegary or even pungent odor is perceived. The ethyl acetate is always synthesized by acetic bacteria as a side product from the formation of acetic acid. The detection threshold is between 150 and 200 mg/L. An odor of nail polish is perceived with concentrations above the detection threshold 82. 2.4.5. Other aromatic defects. Numerous other defective aromas can occur in wines. 81. Excess sulfur dioxide, a substance used in several stages of winemaking, gives wines a smell of burnt matches. Another substance used as a preservative, sorbate, can promote the development of defective aromas described as geranium. Diacetyl, a metabolic product from fungi and bacteria, are usually present in wine in small concentrations. Its butter aroma, if subtle, may be pleasant, and appears frequently in Chardonnay wines. Its concentration above the threshold for recognition brings unpleasant odors to the wine. Ethyl phenols produced by fungi of the genus Brettanomyces /Decara, as quoted above, are responsible for defective aromas described as stable like Stallion and Mouse. 79 Maria Luisa Alvarez Rodriguez et al. Cork taint of wines, role of the filamentous fungi isolated from cork in the formation of 2,4,6-trichloronisol biomethylation of 2,4,6-trichlorophenol. Apple. Environ Microbiol. 2002, 68:5860-9. 80 Jan Margalit and James Crum, editors. Oak Products, Cooperage and Cork. In. Concepts in Wine Chemistry. 2nd ed. South San Francisco, CA, The Wine Appreciation Guild, 2004. p.217-63. 81 Ronald S. Jackson. Olfactory System. In Wine Tasting, A Professional Handbook. London. Academic Press, 2002. p.39-78. 82 Jan Margalit and James Crum, editors. Wine Faults. In Concepts in Wine Chemistry. 2. NDED. South San Francisco, CA, The Wine Appreciation Guild, 2004. p.331-48. 23. Reduced sulfur compounds may be responsible for defective aromas of wine. Mercaptans bring odors of manure or rotten onions, while disulfide presents an odor that resembles cooked cabbage or shrimp. Compounds related with 2-mercaptothanol and 4-methylthio-butanol generate, respectively, intense odors of stable and chives/garlic. 3. Taste and oral sensations. 3.1. Sweetness. The perceived sweetness in wine has its origin in different substances. The main ones are sugars or saccharides, a class of carbohydrates. One of its features is the sweet taste. Among the most simple, there are small molecules with 5 or 6 carbons called monosaccharides. 5 carbon molecules are called pentose while 6 carbon ones are known as hexoses. Monosaccharides can react with each other in a polymerization process, forming larger molecules known as disaccharides, oligosaccharides or polysaccharides, according to the number of monosaccharides composing the new molecule, few or many. Ripe grapes are extremely rich in carbohydrates. The most abundant other hexoses such as glucose and fructose, both with concentrations of 80 to 130 g/L and a small amount of rhamous, 0.15 or 0.4 g/L. Xylose and arabinose pentoses are also found in low concentrations. Glucose and fructose are polymerized, producing sucrose, a disaccharide that is found in concentrations of up to 10 g/L. Vitis vinifera grapes tend to exhibit lower concentrations of sucrose than non-vinifera grapes. Pectin, a polysaccharide, is present with a concentration of 0.2 or 4 g/L. Roughly, glucose and fructose represent 95% of the carbohydrates from the juice of ripe grapes. Hypermature grapes have a higher percentage of fructose. The evaluation of sweetness of these sugars is made by comparison with a 10% sucrose solution used as a standard, arbitrarily set at 100. The sweetest one, fructose, exhibits a relative sweetness index of 115, while glucose, 70. Wine, however, is rich in other sugars, which may not have a sweet taste. Dry wine has less than 2 g/L of residual sugar. The hexoses, glucose and fructose, present in must are fermented by yeast completely, turning into alcohol. These monosaccharides are therefore called fermentable sugars. Residual sugar consists of 24 small amounts of non-fermented pentose and hexose, and polysaccharides known as non-fermentable sugars. Sucrose, which is present in small quantities in must, or added to it in the chaptalization process, is dissociated into fructose and glucose, which are fermented, whereby sucrose should completely disperse. Therefore, the degree of sweetness of a wine depends on the amount of residual sugar after fermentation. Sweet wines are produced with musts that are extremely rich in grape sugars, which do not fully ferment, or their fermentation is interrupted by the addition of a grape spirit, which kills the yeast, or by addition of concentrated grape must, as it occurs in certain German wines. Botrytized wines have a greater concentration of other monosaccharides such as galactose and arabinose. These wines also have another sugar, hexadioluse, formed by the oxidation of fructose. The metabolism of Botrytis cinerea also produces a polyalcohol related to sugars, 2,3-butanediol. Other carbohydrates and derivatives may be present in wine as polysaccharides. Ramogalactranose, mannoproteins, glucans, polyalcohols, xylitol, arabitol, mannitol, inositol, and sugar acids such as gluconic and galactronic acid. These compounds contribute little to the sweetness of the wine, however increase the feeling of wine smoothness and body. 83,84,85. Thus, highly concentrated carbohydrates in ripe grapes are fermented and converted to ethanol. Unfermented residual sugar is primarily responsible for the sweetness of the wine. 3.2. Acidity. Every wine is acidic. Acidity is constitutional part of this beverage, and there are no alkaline types. Acids are essential in wine. They ensure microbiological and color stability and contribute for longevity. And they also determine wine freshness and liveliness. When one says, "This wine is acidic," a more proper way to say it would be, "The sensation of acidity of this wine is excessive for its structure." Thus, for better understanding wine balance, the knowledge of the origins of acidity and sensations generated by it is required. 83. Jan Gallet and James Crum, editors. Must and Wine Composition. In Concepts in Wine. Chemistry. 2. NDED. South San Francisco, CA, The Wine Appreciation Guild, 2004. p.1-55. 84. Ronald S. Jackson. Chemical Constituents of Grapes and Wine. In Wine Science, Principles. Practice, Perception. 2. NDED. Burlington, Ma, Academic Press, 2000. p.232-75. 85. M. Luz Sanz and Isabel Martinez Castro. Carbohydrates. In Moreno Arribas MV, Polo MC, editors. Wine Chemistry and Biochemistry. New York, Springer, 2009. p.231-48. 25. Wine organic acids can come from the grapes or can be formed during the fermentation process. The first are natural organic acids, which develop during the vegetative cycle of the vine. The major ones are tartaric, malic and citric acids. Acids obtained from fermentation are succinic acid, acetic acid and lactic acid. 3.2.1 Acids derived from grapes. Tartaric acid, 2,3-dehydroxybutanoic or 2,3-dehydroxysuccinic, with molecular formula C4H6O6, rarely found in other fruits, is predominant in grapes. It is present in concentrations of 5 to 10 G/L in Vitis vinifera musts, accounting for 1/3 of the acids present in wine. Its concentration in wine is barely affected by the action of yeast. Hydroxybutanoic or hydroxysuccinic acid, also called malic acid, C4H6O5, is present at concentrations of 2 to 4 G/L in wine grapes, depending on the cultivar. Alcoholic fermentation reduces its concentration in 20 to 30% and during malolactic fermentation, this acid is consumed. The name malic originates from the Latin word mala, which means apples, in which this acid is abundant. The sensation produced by this compound is an extreme and aggressive acidity, as seen in some green apples. 2-hydroxypropane 1,2,3-tricarboxylic acid citric acid, of molecular formula C6H8O7 is abundant in the fruit group which is named after it, citrus fruits. Kiwi fruit. Strawberries and raspberries are also rich in citric acid, and in grapes, it is present at lower concentrations. In winemaking, it can be used to correct acidity. 3.2.2 Fermentation-derived acids. Succinic or butanoic acid, C4H6O4, is produced during fermentation and is present in wine at a concentration of 0.4 to 1 G/L. It exhibits a complex, mixed flavor of acidity, bitterness and saltiness. The vinous character of wine is provided by this acid. 2-hydroxypropanoic acid, C3H6O3, also called lactic acid, is generated by malolactic fermentation made by lactobacilli from malic acid, which is more aggressive and harsh to taste. By replacing malic acid by lactic acid, the wine becomes softer and less green. The set of natural and derived acids referred to herein constitutes the fixed acidity of a wine. However, during the fermentation process, low molecular weight acids are produced. Because of this characteristic, these components are volatile and represent another portion of the total wine acidity, volatile acidity, whose main 26 component is acetic acid. Although present in all wines, it must be below the normal threshold of perception, 0.5 G/L. High levels of volatile acidity, which allow detection, are identified as defects. Ethanoic acid or acetic acid, C2H4O2, is produced in small amounts during fermentation. Bacteria of genus Acetobacter convert ethanol to acetaldehyde and then to acetic acid, vinegar. Under specific conditions, these bacteria can also synthesize ethyl acetate from ethanol. Thus, the defect that occurs when there is wine spoilage by Acetobacter is a mixture of smells of vinegar and nail polish. Other acids that make up volatile acids are formic, propionic and butyric. 3.2.3 Measurements of acidity. The total acidity of a wine, TAR, is the total concentration of its organic acids, plus the concentrations of sodium and potassium. The titratable acidity is the sum of the concentrations of titratable protons, measured by titration with a strong base, The titratable acidity is therefore lower than the total acidity. TAR is measured in grams per liter, G/L, and usually lies between 4.5 g and 7 g/L. Because it is the most abundant acid, wine total acidity is usually expressed in tartaric acid equivalents. However, this measure does not provide an accurate correlation with the sensation of acidity of the wine. To better understand this phenomenon, one must understand the concept of pH. One of the characteristic of acids is its degree of ionization in an aqueous solution. In this situation, some of the acid molecules dissociate into two parts with electric charges: a hydrogen atom with a positive charge, and the remainder of the molecule with a negative charge. Free hydrogen ions determine the characteristic of an acidic solution. Each acid has a dissociation constant, Kd, and a number of ionized molecules which defines if an acid is strong or weak. Strong acids are those in which virtually all its molecules dissociate in water, whereas in weak acids, dissociation is small. The organic acids present in wine are all weak. However, there are differences among them. Kd for tartaric acid is 9.10 x 10 to 4, while for malic acid, it is 3.50 x 10 to 4. Therefore, tartaric acid is approximately 3 times stronger, since it produces approximately 3 times more hydrogen ions than the same amount of malic acid. Thus, it also follows that smaller amounts of a strong acid may release the same amount of hydrogen ions than higher amounts of a weak acid. Consequently, the phenomenon that best correlates with our sense of acidity in wine is the concentration of ionized hydrogen, i.e. pH. 27. pH is a measurement defined as the inverse logarithm of the concentration of hydrogen ions. Its scale is usually 0 to 14, with 7 being the point of neutrality. From the definition, it follows that the lower pH values are, the greater the acidity. Because the scale is logarithmic and not linear, the variation of a point on the scale corresponds to an increase or decrease of 10 times the concentration of hydrogen ions and, possibly, our taste of acidity 86,87. Other factors may affect the perception of acidity as the salivary flow rate and the concentration of sodium bicarbonate in the saliva, which neutralizes the acid, the extract of wine, and temperature. The important thing for the taster to remember is that wine is a chemically acidic beverage, and what should be assessed is if the feeling of acidity resulting from the balance between the acids present and other components of wine is appropriate, conveys freshness, and above all, is pleasant. 3.3. Bitterness and astringency. Bitterness, as previously discussed, is a taste sensation which is awakened by coupling the sapid substance to a GPCR of the T2R family. Astringency, in turn, is the feeling of dryness, shrinkage, wrinkling, and roughness of the oral mucosa caused by substances that are capable of precipitating salivary proteins, notably the so-called proline-rich proteins. It is generally accepted that astringency is a tactile sensation detected by the mechanoreceptors of the oral mucosa, although there is some evidence of a specific component of astringent taste. The sensation of astringency is complex, as multiple factors can modify it. Thus, a lower pH tends to exacerbate the astringent sensation. Some tastes, sweet in particular, can reduce the astringent sensation. This Feeling is also influenced by the intensity of individual salivary flow. The role of sensitivity to 6N-propylthiouracil, which is genetically determined, in the perception of astringency is controversial. 88,89. A description of perceived astringency is difficult and imprecise. In an attempt to understand the sensations caused by astringent compounds and standardize their 86 Michael J. Leonard-Lee. Acidity in wine, the importance of management through measurement. Cited 2014 Jan. Available from http://gwi. Missouri. edu/publications/2013 Spring. PDF. 87 Yamar Gallet and James Crum, editors. Must and Wine Composition. In Concepts in Wine. Chemistry. 2. NDED. South San Francisco, CA, The Wine Appreciation Guild, 2004. p.1-55. 88 Martha R. Bajek and Gary J. Pickering. Astringency, Mechanisms and Perception. Crid Rev Food SCI. Neuter. 2008, 48:858-75. 89 Isabel Leschave and Anne C. Noble. Polyphenols, factors influencing their sensory properties and their effects on food and beverage preferences. Jacqueline Neuter. 2005, 81, 1 Supple 330s5. 28. Description, a hierarchical classification of oral sensations aroused by red wine has been developed. 90. Astringency and possible bitterness of wines, especially red wines, are mainly conferred by the so-called tannins, which have great importance in its assessment. However, the sensations caused by the tannins are among the least understood in the organoleptic evaluation of wines. For better understanding, it is necessary to know the phenolic composition of the wine. The word tannin is known and used by all wine tasters. However, this term does not correspond to a defined chemical compound. Its etymology lays on the Gallic word tanno, which means oak. The general definition of tannin is a group of plant extracts, which form complexes with proteins, causing their clotting. These compounds, particularly tannic acid, have been used for centuries for tanning leather. A common chemical feature of these extracts is that they are phenolic compounds. These phenols are extremely important in assessing the wines, especially red wines. Tannins are polymers of simpler phenols. 91. They are responsible for astringency, color and possible bitterness. Such substances are also important for the conservation and ripening capacity of wines. The antioxidant and cardioprotective action provided by wines is exerted by polyphenolic compounds. These compounds are derived from phenol, the simplest of the aromatic alcohols, which have a cyclic carbon ring. The phenolic derivatives present in wine may be simpler or exhibit multiple phenol rings constituting the so-called polyphenolic compounds. The polyphenols present in wine derive from grapes, its stems, seeds, yeast, metabolism and the wood of the barrels or vats. These are usually divided into two broad classes, non-flavonoids and flavonoid. 3.3.1 Non-flavonoids. Polyphenols. Non-flavonoids. Polyphenols are simple phenolic compounds from different sources. For wines, which do not age in wood containers, Its origins are substances derived from hydroxycinnamic acid, which are the predominant phenolic compounds in grape juice and white wine. Hydroxycinnamic acid derivatives are rarely found as free acids in wine. Its usual presentation is conjugated with tartaric acid to form esters. Nevertheless, these derivatives continue to be called acid on the enological jargon. 90 Richard Gore et al. A mouthfeel wheel, terminology for communicating the mouthfeel properties of red wine. Ost J. Grape Wine Res. 2000, 6203-7. 91 Thomas Collins. Tannins, Types and Amounts in Grapes and Wines. In New York Wine Industry. Workshop Proceedings, Geneva, NY, 2–4 April 2003. 29. Its main products are butyric, caphteric and photric acids. These acids are found in the grape pulp, and therefore are present in wine. Although they do not affect taste, they play an essential role in wine color. They are the first to be oxidized, and responsible for the development of brown color in white wines. In small quantities, oxidative acids and its derivatives, butyric and caphteric acids, produce a golden straw color, which is so appreciated in some white wines. Considering wines aged in wood, the most abundant phenolic compounds are derivatives of hydroxybenzoic acid, such as gallic and ellagic acid. These components originated from the hydrolysis of its polymeric forms, ellagitannins and gallitannins, commonly called hydrolysable tannins, which are extracted from oak wood. White wines, which are matured for 6 months in oak barrels, have a level of approximately 100 mg/L. Red wines which are aged for 2 or more years in wood containers, the level reaches about 250 mg/L. 92. Although present in small amounts, another class of phenolic non-flavonoid substances, stilbenes, especially resveratrol, is of great importance for its antioxidant and cardioprotective action. Stilbenes originate exclusively from grape skins, which are produced in response to fungal attacks to vines. For this reason, red wines have higher concentrations of resveratrol, about 7 mg/L. Pink wines usually have concentrations of 2 mg/L and white wines, 0.5 mg/L. 92. 3.3.2. Flavonoid polyphenols. Flavonoids are more complex polyphenolic compounds with multiple aromatic rings. They constitute the majority of red wine polyphenols. The main flavonoids are flavonols, and anthocyanins. Flavin 3,4-diol, leucoanthocyanidins, are present in lesser quantities. Flavanols are the most abundant class of flavonoids in grapes and wine. They are originated from skins and seeds, and are usually called flavin 3-ols. 2 of the common forms of this class are catechins and epicatechins. Oligomers of these 2 substances form procyanidins. These are present in grapes as monomers, but they tend to polymerize in wine, forming the so-called condensed tannins, or proanthocyanidins. 93. 92 Andrew L. Waterhouse. Wine Phenolics. An NY Acad SCI. 2002, 957:21-36. 93 Bausch & Son and M. Isabel Spranger. Review, Quantitative Extraction and Analysis of Grape and Wine. proanthocyanidins and stilbenes. Siengtekvativ. 20:59-89 2005. 30. Flavonols are present in grape skins and apparently play a protective role against UVB rays. The major ones are quercetin, camphorol and the myocetin. Another important group of flavonoids are anthocyanins, which are important in determining the color of the wines described above. Thus, the polyphenols present in wines serve multiple functions. With regard to non-flavonoids, cinnamic acid derivatives, they are vital for the phenomena of browning white grapes must, and white wine. Concentrations found in wines do not contribute to astringency or bitterness. Hydrolysable tannins also rarely affect these characteristics. 94. Antioxidant activity. Some simple phenolic groups, such as catechol and 1,4-dihydroxyquinone, which form polyphenolic compounds present in wine, have the characteristic of being easily oxidized. This property gives them a potent antioxidant action because when oxidized, they protect other wine components from oxygen-free radicals which are quite reactive. Preserving the color of musts is one of the consequences of this protection. The use of sulfites, so too, during vinification, enhances antioxidant activity, since these compounds degrade oxidized hydroquinone, making it available again to capture new reactive oxygen anions. 95. Among flavonoids, flavonols are largely responsible for bitterness and astringency. Polymer catechins and their derivatives, such as procyanidins and condensed tannins, are some of the main sapid substances in red wine. The chemical structure of flavonols also influences the perception of bitterness and astringency. Epicatechin is bitterer and more astringent than catechin. The polymerization of these compounds increases the sensation of astringency and decreases bitterness. Condensed tannins of low molecular weight are both bitter and astringent. Those with high molecular weight have little influence on the appreciation of wines. With age, polymers tend to grow, become insoluble and precipitate. Thus, phenol concentration decreases with wine aging. 95,96. 94. Michael Rentschit al. Non-flavonoid phenolic compounds. In Moreno Arribas MV, Polo MC, editors. Wine Chemistry and Biochemistry. New York, Springer, 2009. p. 509-27. 95. Andrew L. Waterhouse. Wine Phenolics. Ann. N.Y. Acad SCI. 2002, 957:21-36. 96. Isabel Leschave and Anne C. Noble. Polyphenols, factors influencing their sensory properties and their effects on food and beverage preferences. Jacqueline Neuter. 2005, 81, 1 Supple 330 S5. 31. Wine assessment. The attributes of the wine described above are some of the qualities valued when assessing wines. There are different approaches when evaluating wines. The most trivial of these activities, the hedonistic wine tasting by savvy consumers, is individual. In this case, the consumer does not seek to quench his thirst with wine nor to reach the feeling of relaxation and fluctuation from light inebriation. Rather, he seeks pleasurable sensations and wine characteristics that meet his personal taste. For this kind of assessment, there is no concept of right or wrong, or even errors of judgment, since assessment is personal. Another type of wine assessment is the evaluation of the differences between samples and their ranking. Enophilic societies and expert panels in contests and awards usually do this type of tasting. In producing countries with regulation of production of wines and award of certifications such as controlled designation of origin, DOC, AOC, or quality wine produced in a specific region, VQPRD, tasting becomes the wine discriminatory and classificatory instrument. It allows for verification whether the organoleptic characteristics of a given wine meet the specifications required for the grant of a certificate and placing of the wine in one of the legal categories in the growing region. There are still evaluation tastings performed by enologists, producers, middlemen, dealers and traders. The particular tasting performed by a sommelier seeks wine characteristics that provide gastronomic matching with the restaurant dishes. Wine tasting by a wine critic tends to carry the strong bias of subjectivity on his or her personal taste, not always coincident with that of the consumer. The common feature in all these sorts of wine assessments is that they are evaluations conducted through tasting, with greater or lesser degree of technical organization, and usually, with no scientific strictness. The result is expressed by a description, assigning a score, or a rating. Unlike these methods of assessment, there is also wine sensory evaluation, which employs sensory analysis techniques. This discipline is based on a branch of psychology, psychophysics, which investigates the relationship between physical stimuli and the perceptions aroused by them. Sensory analysis is structured and uses knowledge from different fields such as sociology, physiology and psychology itself. 32. The results are subjected to statistical tests to verify its significance. Sensory analysis aims to bring objective responses in relation to food, beverages and other products as to how they are perceived by the people, through their sense organs. 97,98. Sensory analysis assumes the existence of a proper environment, rigorous experimental design, use of methods of effective and descriptive sensory discrimination. The difficulties arising from complex experimental design and the need for prior training of the participants hinder its customary use in the wine industry. 99. However, sensory evaluation is an important tool in the development, evaluation and establishment of food and beverage marketing strategies. Sensory analysis reduces the bias inherent to regular tastings, but does not fully eliminate it. One causes of bias and misconceptions in tasting. Errors in wine assessment have various origins. Physiological and psychological factors are important causes for differences in performance among tasters and, consequently, in their understanding of the wine tasted. The level of knowledge about wines and the degree of training on tasting lead to a wide individual variation in wine assessment. One of the main obstacles preventing the activity of wine tasting to be objective is the language used, so that the sensory perceptions obtained when tasting a particular wine can be communicated to others. 1.1 Physiological factors. 1.1.1 Sensory adaptation. The phenomenon of sensory adaptation has been known for a long time. It constitutes in the specific decreased sensitivity to a stimulus by repeated or prolonged exposure to it. Sensitivity returns with time in the absence of exposure to such. 97 Adriano G. Cruz et al. Sensory analysis, relevance for probiotic, and symbiotic product development. Comparev Food SCI Food SAF. 2010, 9, 4358-73. 98 Jorge Herman Barons. Mini-courses 2010, Fundamentals and Techniques of Sensory Analysis. Regional Council of Chemistry, Region 4, SP. Cited 2014 Jan. Available from http://www.crq4.org. br/sms/files/analyze_sensorial_2010. PDF. 99 Lucy Perrin et al. Comparison of 3 sensory methods for use with the Napping Register trademark symbol procedure, case of 10 wines from Loire Valley. Food Quill Prefer. 2008, 19, 11-11. 33. Stimulus 100,101. In the practice of wine tasting, it is important that a time interval between each inhalation of the aroma and between sips of the same wine is respected. Rinsing one's mouth with water or chewing neutral foods such as bread is also intended to avoid adaptation. 1.1.2 Sensory thresholds and individual variation. Detection threshold is the concentration of a stimulant required for 50% of the components of a human panel to identify the presence of the stimulus, without characterizing it. Threshold of perception or recognition, on the other hand, is the minimum concentration of a substance that allows identification of the stimulus. The recognition threshold is typically greater than the detection threshold. It is important to be aware of the statistical nature of these definitions. 102. Individual sensory variations affect wine tasting from the moment of visual evaluation. If different individuals are requested to point out, under the wavelength spectrum, a pure color, such as pure yellow, it will be seen that different wavelengths 575 nm or 586 nm, will be depicted as pure yellow. 103. There is also an interpersonal physiological variation at perception thresholds to different odorants and tastants. The sensitivity to stimulus intensity for the 4 basic tastes exhibits, likewise, interpersonal variation. 104. These factors produce different descriptions of the same wine within a group of tasters. Some qualities and defects can be notable for a taster and go completely unnoticed by others. A clear example of this phenomenon is the wide variation in the detection of 2,4,6-trichloronisol and in rejection of wines with high concentrations of this compound. 105. 100 Georg von Becchese. The effect of adaptation of the taste threshold observed with a semi-automatic gustometer. J. Gen Physiol. 1965, 48:481-8. 101 Pamela Dalton. Psychophysical and behavioral characteristics of olfactory adaptation. Chemistry Senses. 2000, 25, 4487-92. 102 Jose Aparecido da Silva and Rainier Johannes Antonius Rosestraten. Manual Pratico de Psicofísica. Cited 2014 Jan. Available from page 3. Wikispaces. com/file/view/manual and Sika PDF 103 Philippe Lanthony La perception des couleurs. J. int SC Ivin VIN 1999, 33, Specis 59-62 104 Dewey Unlimited L. Measures of individual differences in taste and creaminess perception. Chemistry Senses. 2008, 33, 6493-501 105 John Prescott. Estimating a consumer rejection threshold for cork taint in white wine. Food Quill. Prefer. 2005, 16, 4345-9 34 1.1.3 Genetic variation 1.1.3.1 Vision. The first characteristic of the wine perceived by a taster is its appearance, and particularly its color. As previously mentioned, dyschromatopsia has a prevalence, which is not negligible, especially in the male population. About 8% of men and 0.5% of women have congenital abnormalities of color vision 106. Thus, the likelihood of a male taster not to be capable of distinguishing shade variations between a young purple, red wine and another more evolved red-orange colored wine is not negligible. 1.1.3.2 Gustation. The perceptual capacity for basic tastes, particularly sweet and bitter, has a significant population variation, whose genetic mechanisms are beginning to be unraveled. 107,108. The genetic regulation of taste perception of some related chemicals, such as phenylthiocarbamide, PTC, and 6N-propylthiouracil, PROP, is well known. And in the general population, there are people with and without the ability to feel its bitter taste. 109. With respect to PROP, a division between individuals able to taste the bitterness aroused by this substance, tasters, was detected. Some of them, supertasters, have high sensitivity to this substance, describing the taste of PROP as extremely bitter. 110. Apparently, the number of lingual fungiform papillae varies for each of these phenotypes, whereby in supertasters its density is higher. 110,111. The most likely gene related to the greater or lesser capacity to feel the bitterness of PROP is the TARS2R38, which features single nucleotide polymorphism, with 3